Welcome to my channel Learnovate. In this video, we will going to discuss about the development of the eye and what are the structures that are derived from the different parts of the brain and different parts of the surface ectoderm. We will also going to discuss when the development of eye starts and what are the structures that are formed at the end of the human eye develop. So the development of eye starts at the 22nd day of the embryonic period, or also called as third week of the embryonic period. At the third week, when the neural system, nervous system was developing and the diencephalon and the telencephalon was differentiating, at that time the optic grooves appear on both the side of the diencephalon by the invagination of mesenchyme, and these grooves are called as the optic grooves. Then what happens, these optic grooves evaginate outward. Here you can see these are the forebrains and these are going to evaginate the optic grooves outward, and these evaginated diverticula of the optic grooves are called as the optic vesicles. They are just like a vesicular form. So that's why this evaginated part is called as the optic vesicle. And we all know that this all area is covered with the surface ectoderm. So what happens, this optic vesicle will going to come in contact with the surface ectoderm. Now this surface ectoderm, that is a primordium of a lens placode, and the optic vesicle, that is a primordium of the retina or the optic cup, will going to contact with each other, and in the next phase what happens, there is a formation of an invaginated groove, and this is called as the optic cup. This optic cup is a double layered cup, and these two layers of the optic cup will ultimately going to develop into the two layers of retina. This optic cup is now going to be attached with the forebrain with a narrow stalk. This narrow stalk is called as the optic stalk. Now here you can see, if we go previously in the optic vesicle, here we can see the connection between the optic vesicle and the forebrain. That same connection is now going to develop into the optic stalk when these optic vesicles form the optic cup. Now we have two structures that are connected to forebrain, that is the optic stalk and the optic cup. The optic cup will going to form into the layers of retina. While here you can see that the lens placode that was formed from the surface ectoderm is now going to invaginate and depend down inside and coming in contact with the optic cup. Now this lens placode has become the lens pit. This lens pit will going to soon invaginate more and more inside, and until that it will going to form the lens vesicle. When the lens vesicle is being formed, the connection of this lens vesicle with the surface ectoderm being lost, and this lens vesicle will going to develop into a lens of the eye, and the surface ectoderm will be repaired again. Now what happens, the final structure would be the development of two layers of retina and the optic stalk. That was the first, that was the optic stalk. Now this will going to form the optic nerve, and that optic nerve is covered with all the layers of the meninges, that is the dura, arachnoid matter and the pia mater. Now this is the layer of the retina that is being formed, and this is the lens vesicle, while this was a surface ectoderm. Now these two structures has been formed, and this is the optic nerve that is being formed that attach the lens and the retina with the forebrain. Now one more important thing, that when this was all developing, there was a groove that appears on the ventral side of the optic cup and along the optic stalk, and that groove is called as the hyaloid groove or the retinal fissure. That retinal fissure contains the vasculature of the retina, and that vasculature that is present in that retinal fissure is called as the hyaloid vessels that contain the hyaloid artery and the hyaloid vein. Now what happens, this retinal fissure is open up to the third week. Now when we go upward and forward, in which we can see the next development of the eye, here in this diagram we can see that that retinal fissure or the hyaloid canal that was open in the third week is now being closed at the week, that is the forward weeks, that are the fourth week and the fifth week, that retinal fissure being closed. And what happens, the blood vessels, that was the hyaloid artery and hyaloid vein, will going to form the blood vessels that are the central artery and the central vein of retina. So they are present in the optic, along the optic nerve, and at the optic stalk they can be formed. So that you can say that the hyaloid vessels will going to differentiate into the central artery and central vein of retina, ongoing development. This is about the vasculature and how the hyaloid canal was closed. Now the last thing that we have to discuss is how the different structures along within the retina and the lens are formed. So now in this diagram we can see when all the structures of eye being formed, coming from the vitreous chamber, the anterior chamber, the posterior chamber, the lens, the cornea, the retina, and the different layers including the sclera, the choroid, all of the structures of the eye being developed at the time when the development of these stages has been complete at the 5th and 6th week. So we all know that the eye is derived from different sources. So what are the different sources and how they combine to form these different structures, we're going to discuss. There are the 3 layers of eye, that are the sclera, choroid, and the different layers, that sclera and the choroid. These 2 layers of the eye are derived from the neural crest cells, while the vitreous body and the eyelids that will going to form in the anterior surface are also going to develop from the neural crest cell. Now the second thing that is going to develop, that is your muscles of the iris. Here you can see this is your iris, and when this is going to develop around the lens or anteriorly where the opening is called pupil, this iris will going to form 2 muscles, that is the sphincter pupillae and the dilator pupillae. They are going to constrict and dilate the pupillary opening. These iris muscles are going to develop from the neuroectoderm. The neuroectodermal layer will form the sphincter and the dilator pupillae, while the neural crest cell will going to form the layers of the eyeball, that is the sclera and the choroid and the eyelid. Then the cornea that is being formed, here you can see that is the cornea, that is the anterior transparent part of the eye. This cornea is being formed, this epithelium of the cornea and the lens, particularly the epithelium of cornea and the lens we have just discussed, they both are developed from the surface ectoderm. So the surface ectoderm will going to form the lens and the cornea, while the different parts are derived from the different type of nerve tissues. So in this way we can conclude that eye is being derived from 4 derivatives: the neural crest cells, the neuroectoderm, the mesenchyme between the surface ectoderm and neuroectoderm, and the 4th one is the surface ectoderm. So when these different ectoderms combine, they form the different structures of the eye, that is the evagination of the diencephalon. And this was about the development of eye that completed at the 5th to 6th week of the embryonic development. So this was all about the embryology, and I hope it will be explained well. Thank you for watching. Subscribe for more. If you have any comment below, like, share and subscribe.